Hey everyone! Today let's dive into one of the monumental breakthroughs in the world of science, the polymerase chain reaction, commonly known as PCR. This technique revolutionized molecular biology and has been a cornerstone in scientific research, forensic science, and medical diagnostics. But how did this come to be? Let's find out. It all started in the mid-1980s with a brilliant biochemist named Kary Mullis. One night, while cruising down a California highway, Mullis envisioned a way to amplify DNA sequences without the tedious processes previously required. This vision led to the creation of PCR, a method that allows us to copy a specific DNA segment millions of times. It's like having a biological photocopy machine at the molecular level. Now, what made PCR a game changer? Before PCR, studying genes required large amounts of DNA, which was not only challenging to obtain but also time-consuming to analyze. PCR, on the other hand, needs just a tiny fragment of DNA to produce enough copies for detailed study. Imagine trying to read a book, but instead of needing the whole library, you just need one page to reproduce the entire content. But PCR wouldn't have reached its full potential without another invention, the thermal cycler machine. This ingenious device automates the PCR process by cycling through the temperature changes required to denature DNA, bind primers, and extend the DNA strand. Essentially, it streamlined the entire process, making it faster, more reliable, and accessible to scientists around the globe. Thanks to PCR and the thermal cycler, what used to take weeks can now be done in hours. This has had profound implications across various fields. In medicine, it's used for diagnosing genetic disorders, detecting pathogens, and even in cancer research. In forensics, PCR helps solve crimes by analyzing the genetic material from a single hair or a drop of blood. And in research, it's indispensable for genetic sequencing and cloning. In conclusion, PCR and the thermal cycler have not just changed the way we do science, they've expanded the very horizons of our scientific inquiry. So next time you hear about a scientific discovery or a breakthrough in medicine, there's a good chance PCR played a part in it. Thanks for tuning in. If you're fascinated by how simple ideas can lead to revolutionary tools, hit that like button and don't forget to subscribe for more amazing science stories. See you next time.